good morning, everyone. And I bring my notes to not forget anything. And please correct me, Michael, if I make a mistake. It's an honor and a pleasure to introduce a distinguished speaker today, Professor Michael Niederweis. He's endowed professor of bacteriology and university professor at the University of Alabama at Birmingham in the United States. He earned his PhD in Germany in microbiology, summa cum laude, in the University of Erlangen, and has since established himself as a leading figure in bacterial pathogenesis, particularly in tuberculosis research. So interesting for many people here. He's been at the University of Alabama since 2004, and over the years he has won numerous awards. It was really hard for me to go through his CV. It's amazing. Over the years, he has won numerous awards, such as becoming elected Fellow of the American Academy of Microbiology, Senior Member of the National Academy of Inventores, and so on. His groundbreaking work focuses on tuberculosis biology, many things, but one that catch my interest is mycobacterial outer membrane biology. And worth highlighting, that I didn't know, is his discovery of purines, like MSPA, which is a beautiful example of fundamental science meeting with technological applications. TB MSPA was a breakthrough that revolutionized nanopore DNA sequencing technology and is licensed to Illumina. So, Michael is a prolific researcher. He has authored over 130 publications in top-tier journals, holds key patents bridging basic science and biomedical innovation. His lab's recent discovery of tuberculosis necrotizing toxin, which hijacks host cell NAD to trigger necroptosis, has opened new avenues for understanding TB pathogenesis. He's also a dedicated mentor, and this is very good for this audience. He has trained an impressive number of scientists, I must say, who are now thriving in academia and in industry. His work is supported by major research grants, including ongoing projects on iron acquisition mechanisms and toxin secretion in TB. Today, he'll share insights from his lab's cutting-edge research focused on the master control of protein secretion in Mycobacterium tuberculosis. So please join me in welcoming Professor Michael Niederweis.
No, I have a thank you very much for the very kind introduction, Alejandro. It's a pleasure to be here, and I also want to thank Martin for accommodating my complicated travel schedule. So I'm excited to be here, and so today I would like to talk about one of our projects, and this started, so it's about the control of protein secretion in Mycobacterium tuberculosis, which is a mechanism which we discovered by coincidence. Because as Alejandro said, we focused initially on just understanding how the outer membrane of Mycobacterium tuberculosis is functionalized. Then we discovered this toxin, which we call TNT, for tuberculosis necrotizing toxin, which is the only known exotoxin of Mycobacterium tuberculosis. And then we wanted to understand how this toxin is actually secreted and exported. And so we stumbled upon the type 7 secretion systems, which have been studied by many other groups for a long time. And then we discovered that everything is actually regulated by a master regulation, which is connected to this toxin. So it's a really, meandering path, figuring out how things work. So very simple questions, but interesting results. So I just want to give you a short introduction, which you probably all know, but it is important to understand how important lipids are for Mycobacterium tuberculosis, because they're not only producing a large quantity of lipids, so up to 60% of the cell dry mass. So usually bacteria have a lipid content of 5 to 10%. But they also produce 10,000 different lipid species, which has been found by lipidomics, and most of these lipids have unknown structures. So we know only like a few dozens by structure, the most important ones, we believe. So, and the reason, I mean, these lipids, the hydrophobic surface of mycobacteria, as you can see here, is the reason why they grow so in funny-like structures on agar blades and form clumps. But more importantly, what we found by cryo-electron microscopy, because, you know, as Alejandro said, we discovered MSPA, this pore protein from Mycobacterium smegmatis. And people, when we published these, our work about MSPA, they said, where can this pore protein be? Because it forms an open pore. It cannot be in the inner membrane, because an open pore would lead to diffusion of all ion gradients, so there would be no energy anymore. So there got to be an outer membrane. So, and based on all these lipids, so we proposed that there is actually, this pore is an outer membrane protein, but we always got the comments back from reviewers, well, it's nice work, but really there is no outer membrane. And I said, eventually, we needed to, I said to my colleagues, we need to show that there is actually an outer membrane. And then this is when we did cryo-electron microscopy, and voila, there is actually an outer membrane. So, and then the next question obviously is, how is this outer membrane functionalized? And we already knew from many studies before, many in the Colorado State University, Patrick Brennan, for example, the lipid composition of all these strange lipids are located in the outer membrane. And they form a membrane which has a very low permeability, but there were no proteins known who would functionalize. I mean, for uptake processes, which are obviously important, secretion processes are also very important, so that none of these proteins have been described. And so my goal is, when we started 20 years ago, to identify proteins in this outer membrane and just see what we can find. So the reason why we focus on these proteins is because this outer membrane has a very different chemical structure than outer membranes of gram-negative bacteria. So very unusual lipid compositions, extremely low fluidity, and most likely when there are novel structures, there are novel molecular mechanisms, and obviously all of these functions here are important for the biology of Mycobacterium tuberculosis. So, but how do you identify these proteins? Because they are not abundant, it's very difficult to isolate them. It's not possible, I forgot to mention this actually, it's not possible to isolate the outer membrane because it's covalently linked to the peptidoglycan. So you cannot separate the outer membrane from the cell unless you destroy everything, and then you have mixtures of proteins. So, and then we took a genetic approach. So it's a long story, and I really don't have time to tell you details of it. But by bioinformatics, combination of bioinformatics and a genetic screen, we identified a dozen or so proteins which we suspected could be pore-forming proteins. So one of those proteins was CPNT, and then we did a lot of studies with the N-terminal domain, which we found so we purified the protein, we demonstrated that it forms a pore, but this pore was connected to an unusual C-terminal domain with no similarity to anything known in the database. So, and the problem was we couldn't clone this C-terminal domain, and it took us almost a year to clone it. We suspected that it was a toxic domain. And since we could not clone this in E. coli, not in Mycobacteria, and not in yeast, we basically tried to eliminate all basal transcriptions in this vector, and we managed to clone it in a eukaryotic expression vector. And then we wanted to test our hypothesis that this C-terminal domain is really toxic. So we used the TET regulation system, which is a tightly controlled system which is used very frequently in eukaryotic expression systems. And so we could express the C-terminal domain in Schurter T cells. And as you can see here, this is how Schurter T cells, cell culture looks like. And when you add doxycycline, you induce expression. And indeed, most of the cells here shown in red are dead, so it's a live cell stain. And the other cells, which are not dead yet, they're on the verge of dying, as you can see. So then we can quantify this. We measured the viable cells after expression of this toxin, and within 24 hours, all the cells were dead. And this is similar when you induce apoptotic cell death by an activated caspase. You have a similar death kinetics. So it's a very rapid cell death when you induce the C-terminal domain of the CPNT protein. So then we tested what function does this toxin have for the biology of Mycobacterium tuberculosis. So that's a macrophage infection experiment. And what you can see here, that approximately 15% or so of the cells of the macrophages die anyway. When you infect them with Mycobacterium tuberculosis, half of the macrophages die. And the CPNT mutant deletion of the CPNT gene reduces the toxicity by 70% compared to the background level. And then you can complement this by putting the CPNT gene back. But when you only put the N-terminal domain back, you get the reduced cytotoxicity of the deletion mutant, which means the C-terminal domain is really the cytotoxic factor in macrophages, so the major cytotoxicity factor in macrophages. So then we did some staining experiments, which I initially believed would not work. So basically what we did is we expressed GFP at that time in TB. So you see here the green bacteria. And then we stained for an antigen 85, which is just a control protein, which is secreted by, it has active function in the paraplasm, but it's also secreted for some reason which you don't know. And then we stand here for TNT using a specific antibody, and you can clearly see that TNT is actually secreted in the cytosol of the macrophages. Here in yellow, you can see some overlap still in the phagosome of the macrophages. So it turns out that TNT is not only the first exotoxin of Mycobacterium tuberculosis, but it has a lot of homologs in other bacteria and also in fungi, all of unknown function at the time. So believe it or not, the toxin discovered in MTB is present in so many other bacteria which are much easier to work with than Mycobacterium tuberculosis. So, but what is the function? Because it had no homologs of known function, so we had no idea. The only thing what we knew that it was toxic in eukaryotic cells, it was toxic in bacteria, so it needed to somehow inactivate an essential mechanism in all of these cells. So then we did an experiment, which the review of our grant said is a really stupid experiment, because we looked in E. coli. We just expressed the toxin in E. coli for 20 minutes before E. coli was dying, and then we did transcriptomics, and we were mainly interested in the genes which were induced by the expression of this toxin. And what we found is that some of the most highly expressed genes in the presence of this toxin were involved in NAD biosynthesis. So then, of course, we measured NAD content in E. coli after expression of the toxin, and there was no NAD left. And the same is when we use the Jurkat T cells, there's no NAD left after expression of this toxin. And then the next question is, because, or at that time was, because a lot of bacterial toxins use NAD as a cofactor, so the ADP ribosylating toxins are very common toxins in bacterial pathogens. So, but they require another factor. They're only active in the presence of a eukaryotic protein. And we wanted to know whether TNT is active by itself or whether it requires a cofactor. And so we did the in vitro experiment. So we purified TNT, and just the protein by itself completely degrades NAD in solution. And when we use an antibody, you can actually see that it completely inactivates the enzyme. So when you look at the operon, you can see here CPNT is the third gene in an operon, and it's preceded by two proteins which have been involved, homologs of these proteins are actually involved in protein secretion, so it was a good guess that they would be involved in secretion of the toxin. And there was one gene of unknown function, is IFT, which we didn't know what the function was, but we suspected that it may have been or maybe an immunity factor. So this was based on a comparison with streptococci, which have a similar system where there is an NAD hydrolyzing toxin, and there is an immunity factor associated with this. So we tested this hypothesis, and lo and behold, so when we did surface plasma and resonance experiments, TNT was coupled on a ship. You inject IFT, so this purified protein, it binds this very high affinity to TNT. And as I can see here, the NAD hydrolysis experiments, it's not only the antibody totally inactivates the enzyme, but also this immunity factor. So it completely inactivates TNT. And this enabled us to purify TNT now in larger quantities in E. coli. Previously, we could only purify a few micrograms of proteins. And now we get really nice bands for the TNT IFT complex. We got 50 milligrams of purified complex. And then we could isolate 2 milligrams of TNT from this complex by heat dissociation. And then Gino Singolani, a structural biologist at that time at the Thomas Jefferson University in Philadelphia, so he solved the structure of the TNT IFT complex. And what you can see here is that the immunity factor binds in this groove, which we showed later, is actually exactly where NAD binds. So it's a very strong complex. So here's initially we did a molecular modeling. So because we only had the structure of TNT and we couldn't get a structure with NAD in a co-crystal. But NAD really fits very nicely into the toxin structure in this groove here. And then we went on and we mutated all the amino acids around this predicted NAD binding site and did the NAD hydrolysis assay. And as you can see, here's the same assay. So if you add TNT, the NAD is hydrolyzed, as you can see here. And then, for example, this 727 phenylalanine has no NAD degradation, and also these 3 amino acids, there's no NAD degradation, which means that these residues are essential for the function of TNT. So here, these red residues shown in red, they are important for the function, for the hydrolysis. So, and then the last experiment we wanted to do in this project was to show that it's really NAD hydrolysis, which is the toxic function in macrophage infection experiments. So when you look at the cytotoxicity, so we just measure in infected macrophages. You see here is a similar experiment as I showed you before. When we use a mutant which is catalytically inactive, it has the same cytotoxicity reduction as deletion of the entire CPNT gene, which tells you that it's really the hydrolysis of NAD which kills the macrophages. So, and then we looked also at NAD concentrations. So when macrophages are infected with Mycobacterium tuberculosis, there's no NAD detectable anymore. The same is when we complement the CPNT mutant, but when we complement with the catalytically inactive TNT, so there is much more NAD left in the macrophages. So the NAD hydrolysis is really the toxicity function of this toxin. So to summarize this part of the talk, is we showed that Mycobacterium tuberculosis indeed produces a toxin which was not known at that time. Everybody believed that because there's a co-evolution of people and Mycobacterium tuberculosis, and TB can be in our bodies for a long time without us getting sick or dying. So many people believe that there is no toxin, but it actually, there is a toxin which induces necrosis by hydrolyzing NAD. It's the major cytotoxicity factor in macrophages, and it's the founding member of a new class of widespread toxins. So then the next big question is, how is TNT secreted? So when you can see here, so here you see an experiment which I showed you before. TNT is shown in red and the bacteria are shown in green. they're still trapped in the phagosome at this point of the infection. But what was remarkable that CPNT does not have a signal peptide. You know, all the outer membrane proteins in gram-negative bacteria, they have usually a sex signal peptide. So CPNT did not have this. So we had no idea how CPNT would actually get to the outer membrane. And we also didn't know how TNT would actually secrete it, or whether it's the entire protein which is secreted. So I already mentioned the two preceding genes of the CPNT gene, the ESX-ENF genes. So they are homologs of these ESX proteins, which are encoded in all the type 7 secretion systems of Mycobacterium tuberculosis. So these so-called ESX systems, they are the only known protein secretion systems in Mycobacterium tuberculosis. And they range from very simple configurations here with the ESX4 system to very complicated systems like the ESX1 system. And not all pathogenic mycobacteria have all of these ESX systems. So there are the important differences. So the question was, one of the questions was, which of these ESX, so first of all, is CPNT dependent on an ESX system, and then which ESX system is actually responsible for secretion of TNT? So here you see again the ESX F and E genes in front of CPNT, and they do have a sequence similarity to this motif, this YXXS, the aspartate glutamate motif, which is so Wilbert Bitter in Amsterdam, and his group, they showed that this is a secretion signal for the type 7 secretion systems. And these proteins are homologs of these secreted proteins. And then we found that CPNT also has 3 of these motifs. So we mutated all of these motifs. I don't have time to show you this. But when we mutate, for example, this putative secretion signals, the protein is completely gone. There's no expression anymore. It's probably not secreted and then degraded. So when we mutate this motif, only TNT is not accessible on the cell surface anymore, but the protein is still detectable. So then we wanted to know, what is the role of the ESX-ENF system in secretion? So what we did is we deleted the entire CPNT operand from Mycobacterium tuberculosis, and then we complemented just with the CPNT gene and the immunity factor gene, and then with the entire operand. And what you can see here is when you add the entire operand, you see nice expression of TNT. And when you only add CPNT and the IFT, there is no expression or no TNT on the cell surface detectable anymore. So these are whole cells, and we just put antibodies on it and do fluorescence microscopy. So we can do the same experiment more quantitatively using flow cytometry. So what you can see here is, so this is the background, and so when we add just CPNT IFT, you see a minor change, but you see a much better, much more quantitative shift to more fluorescence when we using a TNT antibody here when we add ESX-ENF, so which confirms the microscopy data that ESX-ENF are required for secretion of CPNT and TNT on the cell surface. So then we did the same experiment in infected macrophages. As you can see here, wild-type MTB, you can detect TNT is secreted. So this is the CPNT operand deletion mutant. There's no TNT detectable anymore. We just put the CPNT back. There's no secretion. And when we put the entire operand back, just ESX-ENF in addition, there's full secretion of the operand. So confirming that ESX-ENF are really required for secretion of TNT. So then we wanted to understand, how does ESX-ENF actually, which role do ESX-ENF play in secretion of TNT? So we did a very simple experiment. We isolated Mycobacterium tuberculosis, did subcellular fractionation, And then we just separated membrane fractions and soluble fractions. So this is a control protein for soluble fractions. That's a membrane fraction. And you can see that both CPNT and ESXENF are in both fractions. So they're both membrane associated, and they're probably on the somewhere in the periplasm, maybe on the transition. And what was really surprising is when that we were able to detect ESXF on the cell surface of in vitro grown Mycobacterium tuberculosis. We could not detect ESXE, as you can see here. So ESXF is actually on the cell surface, which means it's membrane associated, so it means to be anchored in the outer membrane of Mycobacterium tuberculosis. So then we did some in vitro experiments. So we looked at just liposomes, and we stained these liposomes with FM464. That's just a hydrophobic dye. You see in fluorescence microscopy, so you see a nice red color. And then we incubated these liposomes with purified ESXENF, and what you can see here is that these liposomes are strongly deformed in the presence of ESXENF. And then we did planar lipid bilayer experiments. That's the technology which Alejandro briefly touched about. So we worked a lot with pore proteins before, MSPA, for example. And we wanted to see whether ESXENF actually form pores. And lo and behold, what you can see here is a membrane. There's no current flowing over the membrane when there is no protein inserted. And when there's a pore protein inserting into the membrane, so this is an electric field, the membrane is in the middle. And when you have put two electrodes on each side of the membrane, you can measure the conductivity of the membrane in electrolyte. So we use usually potassium chloride, 1 molar. And when there is conductivity for each pore which inserts into the membrane, we see an increase of the conductivity. So it's like it's a really sensitive technology where we can detect individual pore proteins. And then we quantify this. So this is the conductance of the pores, and then you can plot the number of insertions, and you can see that there are many insertions which we can detect with purified ESXENF, which means that these proteins form pores in the outer membrane, but also that it's really strongly pH dependent. So when we reduce the pH, at low pH, there's almost no pore formation anymore. So this is related to the biology of MTB. We can later discuss about this, but yeah. And then we were interested in structures. I told Alejandro we are really interested in structures. We collaborate a lot with structural biologists. So this was work done by Terje Docklands, who is an electron microscopist at UAB. So we looked at purified ESXENF, 20,000 particles. So this is not a high resolution. It's not cryo-electron microscopy, so it's negative stain. But the reason why we use negative stain is because it's easy to see pore proteins. Because when the pore is filled with uranyl acetate, then it's water filled, and then uranyl acetate accumulates in the pore, and what you can see here is a black dot. So it's a really nice way of detecting a simple technology to visualize pore proteins. And lo and behold, ESXENF form pore proteins. You can easily see here. And then you can look at different particles. You get better resolutions by making classes of these proteins. And what you end up with is like a somewhat higher resolution model. And here the ESX is a model of the structure of ESXENF or homologs which fits very nicely. It's a pentameric structure which forms a pore. Here's a side view of the same. So what was interesting is when we looked at the interface between ESXE and ESXF, there are these highly conserved tryptophans, and we were wondering, or it looked like that they play important roles in the interaction between these two proteins. So what we did is we made, here you see the same model again where these tryptophans are located. So one is in ESXF and two are in ESXE. And so we mutated these tryptophans, and when we did this, we completely abolished the pore-forming activity. So association, I mean, at least what we can say is that these tryptophans are really important for pore formation of these proteins. And these mutants are very interesting because you can actually use these mutants to probe whether pore formation is biologically relevant for these proteins. So this will come up later. So when we tested these pore-forming proteins with these tryptophan mutations, they don't do anything anymore to the liposomes. So you can quantify the number of deformed GOVs. So these are gigantic unilamellar vesicles. And you can see there's a significant difference compared to the wild-type protein. So then the next question was, which ESX system out of the 5 known in Mycobacterium tuberculosis secretes TNT? So, and then we made mutants of Mycobacterium tuberculosis. So we actually, for most of these experiments, we use an avirulent strain of Mycobacterium tuberculosis, which was developed by Bill Jacobs at the Albert Einstein Institute in New York. So here that's a control. This is the CPNT deletion mutant, and then we deleted all the ESX systems or made them unfunctional. So ESX1, ESX2, ESX4. I didn't show you the data for ESX3 and 5 because they had no phenotype. But what you can see here is obviously in the wild type you can detect TNT. You don't see this in the CPNT deletion mutant. Obviously, when you delete the ESX1 system, you can detect TNT, so it has no role for secretion of TNT. So ESX2 is the same, it's like wild type, but ESX4, no detectable TNT. So it's really the ESX4 system. We tested ESX3 and 5, and there was also no phenotype. So we quantified this, and as you can see here, the lack of ESX4 has the same phenotype as deletion of the entire CPNT gene. So we quantified this here using flow cytometry. You can see here, that's the TNT surface detection, the fluorescence, and you see you delete the CPNT gene, there's less fluorescence, there's no change here, delete ESX4. This is similar to the CPNT deletion mutant. So this is an analysis of 50,000 cells by flow cytometry. And when we put the ESX4 upon back, you actually overexpress TNT on the cell surface. So then we wanted to understand what is actually important for TNT trafficking. So what is the mechanism how TNT actually gets access to the cytosol? So for this, the phagosome, the bacteria are trapped in the phagosome, which is a membrane engulfs the bacteria, and they cannot get out initially out of this phagosome. So they need to somehow permeabilize the phagosomal membrane so that other proteins, like for example kinases or the toxin, have access to the cytosol. So the way how what was actually known well before we started doing this work is that the ESX1 system is very important for this. As you can see here, wild type really secretes TNT very efficiently into the cytosol. This is the CPNT deletion mutant, no detection. ESX1, no detection. And this was known before, that ESX1 is really important for Mycobacterium tuberculosis to permeabilize the phagosomal membrane. But when you look at, so these are control experiments here. But then when you look at the ESX2 operon, the ESX2 system, there's no secretion of TNT into the cytosol, and the same with ESX4, which you would expect because it doesn't even get to the cell surface of MTB, right? And then, so we wanted to know whether the ESX4 system is also involved in phagosomal permeabilization, which we cannot test using TNT as a marker, obviously. So for this, we used another method. So we used galactine-3 staining. So galactine-3 recognizes glycans which are only released into the cytosol of the macrophages when there is membrane damage. So it's a membrane damage marker. So as you can see here, wild-type Mycobacterium tuberculosis damages the membrane as expected. ESX1 system does not, as we also expected, but also ESX2 and ESX4 don't do this. So really ESX4 also plays a role in damaging the membrane. So it's an effect which goes beyond just secretion of TNT. And we can complement these phenotypes by putting the ESX systems back. So our model at that time was, okay, so now we have the CPNT operon, which is induced at some point. And then ESXE and F are secreted, that's step number 1, get exported by the ESX4 system. ESXE and F form a pore in the outer membrane, and then CPNT is somehow recognized by the type 7 secretion system by ESX4 by an unknown mechanism. We still don't know how this works. And then it's exported to the outer membrane, and then CPNT forms a pore in the outer membrane. That's how we discovered the protein. Then TNT is translocated to the cell surface, and then it's secreted. And this is what happens in the macrophage. So the macrophage takes up MTB, it's phagocytosed, and then it's trapped in the phagosome. And then TNT is secreted by the ESX4 system, including ESXE and F, including the ESX1 system, and help also from ESX2 and ESX4 permeabilizes the phagosomal membrane, and then TNT and other virulence factors can actually leave the phagosome. And TNT kills the macrophage by necroptosis, which is a totally different story, which I don't have time to tell you. It's actually very interesting. So NAD depletion induces necroptosis. This destroys the macrophages, and then MTB can disseminate. So that's the mechanism how TNT can exert its toxic function. So then the next question is, how does the ESX4 system enable CPNT and ESXE and F export? So this is ESX4, luckily, is a very simple system, so there are not too many proteins involved, and all of these proteins shown here, they form part of the inner membrane complex of the type 7 secretion systems in Mycobacterium tuberculosis. So what was not known is the function of this protein. Has no homologs of known function. And the ESX U and T proteins, they are homologs of the other. So this, by the way, is ESXA of the ESX1 system. This is ESAT6, which plays an important role in, it's an early antigen secreted by Mycobacterium tuberculosis, plays an important role in diagnosis of bovis and TB. So yeah, and then we focused initially on ESX U and T. We wanted to know whether they may actually play a similar role as ESXE and F in secretion of CPNT and the toxin. So first we did a control experiment. So this is, we did other experiments in vitro, which I didn't have time to show you, but these are the macrophage infection experiments. So we just looked at secretion of ESAT6 or ESXA, as it is called now. And as you would expect, when you have a ESX1 deletion mutant, you don't, here when we detect ESAT6, ESXA, you see lots of protein here in the cytosol, almost no protein in the ESX1 mutant, and when we complement this, there's a lot of protein here. So, and then when we looked at ESXU, we only have an antibody for ESXU. So you can see here that ESXU is also secreted into the cytosol of the infected macrophages. So it's not only, it's an effector protein like you would expect, but the same is true for ESXF. So when you see here, when we look for ESXF in the infected macrophages, you see ESXF signals in the cytosol of the infected macrophages. When you delete CPNT, there's no ESXENF, and we can complement it, and you get this back. What was really interesting is when you look at the ESXUT mutant, there is almost no ESXENF left detectable. And when we complement ESXU and T, you overexpress basically ESXF. And this was really surprising that they seem to be dependent on each other for secretion. So, and then we did in vitro experiments where we just looked at CPNT assembly in the outer membrane staining, by detecting the surface accessibility of CPNT. And surprisingly, when we delete just the ESXU-T genes, there was no TNT detectable anymore. So ESXU and T are actually required for TNT secretion. And we can quantify this as shown here by flow cytometry, and this is the quantitative analysis of the microscopy, fluorescence microscopy. And as you can see here, deletion of ESXU and T has the same effect on TNT secretion as deletion of the CPNT gene. So, and then we did similar experiments previously as we did previously for ESX-ENF. I mean, we found that ESXU and T, we did subcellular fractionation experiments using the ESXU antibody, and it's so we only can detect really when we overexpress ESXU-T, as you can see here, and you can see it's both in the membrane fraction and in the soluble fraction. And ESXU is also detectable on the cell surface, as you can see here. So, and then we tried to quantify in vitro whether it's really true what we saw from the macrophage infection experiments, whether ESXU and F are really codependent on each other for surface localization. You can see when we, again, for ESXF and the UT mutant, we cannot detect ESXF on the cell surface. And when you do, I don't have the fluorescence microscopy data for ESXU here, but what you can see here in the flow cytometry experiments, when we detect ESXU, you can see here, this is the wild type. You delete ESXU, it's not so detectable anymore. You put it back, you get wild type levels. And then when you look at the CPNT operant deletion mutant, there's not ESX-ENF, and you lose detection of ESXU. So it's really, and then we get the same effect when we have the non-functional ESX-ENF mutants. So it's really ESXU and T and ENF seem to be dependent on each other. So we wanted to do an independent experiment to see, so the hypothesis was that they may interact with each other. So we used E. coli, we co-expressed ESXU and T and ENF in E. coli, and then it's where we use, for example, ESXT as a bait using histidine tag, and then we use different tags for detecting ESXU, F, or E. And what we can see here in the elute, where we only have ESXT, we can also detect ESXU and F and E. So they all interact together. So obviously this could be an artifact because this is like expression in E. coli, so it has nothing to do with mycobacteria. So then we asked ourselves whether it's possible to detect this complex in Mycobacterium tuberculosis. And so we did a co-localization experiments using fluorescence microscopy. And here you see bacteria in green. And then using antibodies, we can detect ESXF, we can detect ESXU. And then we can, when we overlay them, you can actually see here, this is all of them merged together. You see that they co-localize in MTB on the cell surface of MTB. So we have a more than 60% co-localization frequency of ESXF and U. So yes, these proteins are together on the cell surface of MTB. So, and then, so now let me back up a little bit. So so now we know that both ESXU and T and ENF interact with each other, and both of them are required for secretion of TNT. So then we ask ourselves, maybe TNT is not the only protein which is dependent on ESXU and T and ENF for secretion. And then we did a very simple experiment. This is what I just showed you. We tested the secreted substrates of all the other ESX systems. So ESA6 or ESXA for the ESX1 system, D for the ESX2 system. So this should be H. So sorry. So this is copy mistake. So this is ESXH and ESXN. So for all of these other ESX systems, we did the same experiment. We just wanted to know whether the mutant which does not have ESX-ENF or a mutant which does not have UT is actually capable of secreting these substrates of the different secretion systems. And what you can immediately see, we lose detection of almost all of these proteins when either ESX-ENF or ESX-UT are missing here. So obviously this is not a quantitative experiment. We did flow cytometry. We basically did the same, obtained the same results. So here this is the HA tagged ESXA is nicely detectable, and when you miss either ESX-ENF or UNT, we lose the signal. So it's the same for all of these proteins. So yeah, and then we came up with this model, which of course is kind of a heresy in the TB field. Pretty much everything what we do is, initially, at least. So yeah, here you have the CP interoperon, which encodes ESX-ENF, which, as I showed you before, is secreted by the ESX4 system. So ESX-ENF form a pore in the outer membrane. But now we know that they actually interact with ESX-UNT and form a pore. So these systems are closely coordinated. And the establishment of this pore or this secretion system is required somehow for secretion of most, or if not all, of the other ESX proteins of the other type 7 secretion systems of Mycobacterium tuberculosis. Of course, this is really heresy, because most people in the TB field think that they have separate functions, which is true, because ESX1 is very important for virulence, for phagosomal permeabilization. ESX2 has no function or no known function. ESX3 is required for iron utilization, and ESX5 is required for secretion and export of a lot of outer membrane proteins of Mycobacterium tuberculosis, in particular PPE proteins and PE proteins. So now we claim that everything is dependent on these two systems. Okay, and then we say, well, before we publish this, we better make sure that this is really true. So we did some control experiments. So one control experiment is our data for the ESX proteins are really good. However, there are many other proteins secreted by the ESX systems. For example, ESPB has been studied a long time, and it's a proven substrate of the ESX1 system. And then our question was, is ESPB secreted in the by Mycobacterium tuberculosis if ESXU and T or ENF are missing? Yeah, and here's the result. So we did a macrophage infection experiments, because this is the most closely related to the real biological environment of MTB. So we HA tagged ESPB, and we can nicely detect it as being secreted. So we confirm the previous publications. When ESX-ENF is missing, we can't detect it anymore. And the same is when UNT is missing. So really, that's another substrate by another unrelated ESX system, which is dependent on ESX-EF and UNT. So then Alejandro briefly managed. We also mentioned this in the introduction. We have been working on iron utilization, and we found that PP62 is an outer membrane protein which is required for heme uptake. So we know that PP62 is dependent on the ESX5 system. So we wanted to know whether PP62 is also dependent on these small, on ENF and UNT. And lo and behold, we did the experiment. So that's an in vitro experiment. Is when ESX-ENF are missing, we can't detect PP62 on the cell surface anymore, and the same is with UNT. So now we have two additional unrelated proteins which are secreted by other ESX systems of MTB, and they all depend on ESX-ENF and UNT. Obviously, this is a small selection of substrates, but this seems to confirm our model. Yeah, and then we showed in a last experiment that ESX-ENF, and DC and UT are also required for phagosomal permeabilization, as you can see here. Okay, just to summarize, I hope I didn't bother you too much with all these ESX, EF, whatever, UT. Yeah, to summarize this, is we discovered that TNT is the major exotoxin of Mycobacterium tuberculosis and kills infected cells by NAD hydrolysis, which then induces programmed cell death, which is called necroptosis. So we show that the type 7 secretion system ESX4 and the forming ESX-ENF proteins are essential for CPNT export and TNT secretion, and ESX-UT I should actually add here. And then the ESX1, 2, and 4 systems are required for phagosomal permeabilization and TNT trafficking, not only TNT, but in general for all proteins of MTB, which are secreted into the cytosol of infected macrophages. And these small ESX proteins have dual functions. They not only are in the outer membrane of MTB, but they are also secreted substrate proteins. So that's why we can find them both in the water-soluble fraction, but also in the membrane fraction. And ESXU and ENF and UNT interact with each other and control secretion of all the other ESX systems. So I need to say that this is not an absolute control, of course not. I mean, there's always basal secretion by the other systems, but if you really want to see full secretion activity, it appears that these proteins are necessary by all the other ESX systems. So this is my group. Most of the work on this project has been done by Rashmi Ravanindra-Nair, she is here, and with the big help of Virginia Mekle, which is my wife, and Anna's cousin. And Mikhail Pavlinok is a long-term, he has been working with me for 20 years, and he does all the lipid bilayer experiments. And David Paruela Games, he was a postdoc from Spain. He returned to Spain, has a really good position there. And yeah, he did all the necroptosis experiments, which I unfortunately couldn't show you. So yeah, I want to thank all of these collaborators and also Bill, Roland, and Mireille for reagents and funding. And I want to thank you for your attention and remind you that all models are wrong, but some of the models are useful. So thank you for your attention.